we're going to have some more people still trickling in when they realize we're not in MDCL 3023. But thank you everyone for coming today. It gives me really great pleasure today to introduce our speaker, Professor Colin Kretz. Simply because sometimes we don't recognize the excellence that is just down the street from us, just because they happen to be working on a slightly different topic. But I'm really proud, actually, that Colin is here at McMaster. So Dr. Kretz is an associate professor in the Department of Medicine and a member of the Thrombosis and Atherosclerosis Research Institute here also at McMaster. And he joined MAC in 2016 following postdoctoral training at the University of Michigan. He runs a basic science research program, a very sort of innovative and exciting one that uses high throughput screening tools coupled with traditional techniques in protein biochemistry to try to understand the regulation of proteases in the cardiovascular system. Many of you will recognize that these proteases have very important roles in cancer as well. And we're really glad to have Dr. Kretz here today. I think you guys will recognize a lot of overlap. And, you know, one of the themes that we've all been talking about at the CDCR recently is exploring a lot of important interdisciplinary and multidisciplinary collaborations. So his current research focuses on the ADAM TS13 von Willebrand factor axis. And for this work, he receives funding from both CIHR and NSERC. So it gives me huge pleasure to welcome Dr. Colin Kretz. Thank you, Colin, for speaking to us today.
Well, thank you, Sheila, for the kind introduction and for the invitation to speak to you guys today. I did my best to come up with a provocative title to my presentation, and I hope that you don't feel like this is a bait and switch where I sort of go down the hemostasis rabbit hole too far. But I think that some of the technologies that we do work on to answer questions in the cardiovascular space can be widely applied to other fields. And so I wanted to have a broad title to my presentation in the hopes of not scaring too many people away, and hopes that we could get enough people in the audience. So thank you everybody for coming. It's a pleasure to be here. So there is a little bit of biology that I did want to introduce you all to before we get into the meat of the project. And my lab and actually many of the labs at TaARI are primarily focused on hemostasis, which is the formation of a blood clot in response to a blood vessel injury. And blood clots are very complex and dynamic structures that involved coordinated responses between circulating blood cells like platelets and other immune cells that are recruited to sites of blood vessel injury, along with the activation of the coagulation cascade that deposits an insoluble fibrin matrix. And it's the convergence of both these soluble cells and these cells and these insoluble coagulation factors that lead to a rapid response to blood vessel injury. Many of you may know that defects in the coagulation system can lead to common inherited and acquired disorders, many of which are a result of chronic illnesses. And the hemostatic system evolved to balance between bleeding and thrombosis, which is in order to maintain the fluidity of blood so that the blood can deliver oxygen and nutrients to various organs and tissues in the body, but also rapidly respond to disruptions in the cardiovascular system. And so defects in the pathways that limit blood clot formation can lead to thrombosis, and defects in the procoagulant activities in the blood can lead to bleeding. And for example, bleeding can be soft tissue bleeding, like in platelet disorders, or in the hemophilias, you can get musculoskeletal bleeding into the joints. And then the most feared bleeding condition is hemorrhagic stroke, where you get intracranial bleeding. And on the other side, on the thrombosis side, we often talk about pulmonary embolism, ischemic stroke, but we also have conditions that are a result of chronic illnesses like diabetes, such as peripheral vascular disease, which can lead to limb amputation. This is a result of microvascular thrombosis. And then there's these paradoxical conditions in the blood clotting system where you get disseminated activation of the coagulation system that leads to a consumption of all your clotting factors, and you get this bleeding condition as a consequence of this. But this is the result of thrombosis. So my lab focuses on two key mediators of the blood clotting system, which is von Willebrand factor and ADAMTS13. So von Willebrand factor is the largest protein in your body. VWF is a multimeric protein, and it exists in circulation in this sort of globular form. And under conditions of blood vessel injury, these large VWF multimers bind to the subendothelial matrix and snake out into the circulation, where their role is to capture platelets and recruit them to the site of blood vessel injury. These VWF platelet strings, that they're called, can grow to be millimeters in length and can rapidly occlude your blood vessels. After platelet recruitment and aggregation, the coagulation cascade deposits a fibrin matrix. So because VWF is this multimeric protein, this VWF platelet string aggregate can continue to grow and will rapidly occlude your blood vessel if it's not adequately regulated. And that is the role of ADAMTS13, which is a metalloprotease that specifically recognizes these ultra-long VWF strings and sort of this shear-dependent mechanism and cleaves them, and in this way sort of prunes the blood clot to keep it from overgrowing and occluding your blood vessel. And so ADAMTS13 is an anticoagulant protein, and von Willebrand factor is a procoagulant protein. And so understanding the dynamics between the regulation of ADAMTS13 and von Willebrand factor is important to understand the dynamic balance in the coagulation system. So just a little bit more on von Willebrand factor. It is a shear-dependent protein. It's sort of some very interesting biochemistry that if you're interested in this sort of stuff, I'd encourage you to read up on. Under low shear or venous conditions, VWF plays a small role in blood clot formation. It plays a dominant role under arterial shear rate conditions, where that shear rate is required to activate the protein in order for it to bind to platelets. And von Willebrand factor is pathogenic at very high shear rates, such as at the site of atherosclerotic plaque lesion. And so regulating von Willebrand factor is important in understanding new treatments for cardiovascular diseases. Von Willebrand factor also plays an important role in inflammation, and this is my token immunology slide for people in the audience. So von Willebrand factor is released from activated endothelial cells and plays an important role in the recruitment of immune cells to sites of vascular inflammation and vascular damage. And it does this either directly binding to receptors on immune cells or indirectly by the recruitment and activation of platelets, which then release or decrypt receptors that will recruit these immune cells. And so in many ways, regulating von Willebrand factor it could be a universal target to treat inflammation and thrombosis. And certainly this is what I say in my CIHR grant applications. So a little bit about ADAMTS13. This is the protease that my lab spends a lot of time thinking about. This is a very large protease. It's 180 kilodaltons. It's the primary molecular regulator of von Willebrand factor. And the cleavage of von Willebrand factor by ADAMTS13 is dependent on shear forces. That shear force applied to a VWF multimer actually unfolds the domain that contains the cleavage site, and it's that unfolding that exposes the cleavage site, and ADAMTS13 can cleave it. And this interaction between ADAMTS13 and von Willebrand factor requires extensive exocyte interactions in order for the protease to recognize and dock to its substrate. So this is a very dynamic interaction that that physicists have been studying and engineers have been studying for understanding the mechanobiology of von Willebrand factor. Makes it a nightmare for a protein biochemist like myself to study it, but it is ripe for new discoveries in the regulation of protease biology. So ADAMTS13 itself is a fairly enigmatic protease, and this is sort of where our story begins. It is secreted as an active protease and exists in circulation for 4 days. And for those of you who know anything about plasma and blood, it is chock full of protease inhibitors, and it's designed to inhibit proteases very, very rapidly in order to localize a blood clotting response to the site of injury. You wouldn't want an activated clotting protease to float away due to flowing blood and cause blood clotting elsewhere in the circulation. And so when we think about cardiovascular proteases, we're constantly talking about balancing the rates of activation or release with the rates of inhibition. And ADAMTS13 violates all of these principles because it's always active and it's always around. So this leads to some interesting questions. If it's constitutively secreted and constitutively active and has this really long half-life, how does it maintain its activity to von Willebrand factor while still resisting inhibition by these broad spectrum metalloprotease inhibitors that exist in blood? So this is an interesting sort of protease biochemistry question. And despite the sort of seemingly unregulated proteolytic activity, it only recognizes von Willebrand factor. So it has incredible specificity for its substrate. So the goal of the first part of my talk here is to explore some of the ways in which we have tried to unravel the regulation of this fairly unique protease. So the big question that we're going to try to address here is, what are the mechanisms that regulate ADAMTS13 activity? And so to address this question, we've turned to an ancient technique called phage display, but we've put sort of a new spin on this. And during my postdoc, this was during the emergence of next generation sequencing, and we were looking for creative ways to adapt this new sequencing technology to old tools. And so many of you may know that phage display is the old technique where you display a protein of interest or a library of proteins of interest on, for example, a filamentous bacteriophage. And in principle, what this does is it provides a physical link between a protein and the DNA that encodes that protein. And this is a very simple particle. It's probably among the simplest particles with which you can achieve this. And in traditional phage display, you relied on the replicative nature of the bacteriophage particle, but now we don't need to replicate it because we have the DNA attached to it. So in substrate phage display, we display a random peptide library on the surface of the bacteriophage, which is flanked by an epitope tag. And the principle here is that when you immobilize this phage library to beads, for example, and expose it to a protease, only the phage that are displaying a peptide sequence that can be cleaved by that protease will be released. And because the DNA that encodes that peptide comes with the phage particle, we can perform next generation sequencing analysis on the released phage to understand what are the properties of the proteins that, that were cleaved. And this provides incredible amounts of depth to our protease, to understanding substrate recognition determinants for proteases, far beyond the dozen or so peptides that you would have gotten from traditional phage display approaches. So the first test case that we chose to use, which made many of the cardiovascular biologists sort of roll their eyes, was thrombin. The reason we chose thrombin is because it is the most heavily characterized protease in the human genome. It's a serine protease that's intimately involved in the blood clotting system and is very, very well-defined substrate recognition. And so when we applied our deep protease profiling approach to thrombin, we identified over 6,700 cleaved peptides. The vast majority of these peptides contained amino acids that we know thrombin likes to recognize and cleave. So for example, the canonical substrate recognition motif for thrombin is proline, arginine, serine, and we see that in our substrate recognition motif here. But what this technique also allows us to do is to understand what are the determinants that antagonize cleavage for thrombin, for example. So what we were able to uncover here is that certainly thrombin will cleave at arginine, but it won't cleave at every arginine like, let's say, a trypsin does. Thrombin will not cleave at arginine if it's flanked by acidic amino acids, for example, or by another arginine residue. And so by using this sort of deep protease profiling approach, we're able to comprehensively characterize the substrate specificity determinants of proteases, and even for something like thrombin, understand new and interesting properties of that enzyme. So we thought, well, let's apply this now to ADAMTS13. Why is it that ADAMTS13 only recognizes von Willebrand factor? Maybe it's evolved exquisite specificity for its substrate, and it's just not capable of recognizing anything else. And so when we exposed ADAMTS13 to this library, we got 96 peptides out of our screen, but that really isn't anything. In fact, many of these were chalked up to statistical anomalies. So why is it that ADAMTS13 was unable to cleave any of the 64 million unique peptide sequences in this library if it's a constitutively active protease? So one hypothesis that we had is, well, maybe it can't directly gain access to the active site unless exosites are available, which would be sites sort of removed from the actual active site. So what we decided to do next is insert our library into a minimal biochemical substrate for ADAMTS13, which is derived from that domain that is shear activated in von Willebrand factor. And this is a 73 amino acid peptide that is known to bind to other regions on ADAMTS13. But we've randomized the P3 to P3 prime residues, which is right around the cleavage site. And when we exposed this new library to ADAMTS13, we immediately identified over 1,500 significantly cleaved peptides, suggesting that for some reason, the ADAMTS13 active site is cryptic unless there's an extended substrate binding interface. When we analyzed the peptides that we identified from this screen, we were thrilled to see that the wild type amino acids for von Willebrand factor within this interval were among those 15 or 1,600 significantly cleaved peptides, but was not the top ranked peptide. So what this tells us is that ADAMTS13 does not have exquisite specificity for its protease, that it can recognize and cleave other substrates, but that it is this extended binding interface that allows it to dock to substrates at the cleavage site. So we identified the substrate recognition motif for ADAMTS13, which is largely dominated by a leucine at position P3 and large bulky aliphatic amino acids throughout the substrate. So, that observation from this deep protease profiling approach allowed us to go on and make other discoveries about ADAMTS13 substrate specificity. So in a paper that was just recently published this year, Kanwal Singh, who just graduated with his PhD, was able to identify the features around the active site of ADAMTS13 that maintain this latency. And he found that features around this calcium binding loop, this variable loop, and this weird gatekeeper triad, which for the purposes of this talk are not super important, but also this weird hydrophobic patch in the active site play an important role in keeping the ADAMTS13 active site cryptic and only becomes opened in the presence of its substrate. And we've sort of coined this term called substrate-induced activation of ADAMTS13. And that's largely caused by these autoinhibitory modules. So that was sort of our first foray into this topic, which we've sort of called deep protease profiling. But we're hoping to expand the application of this technique into the study of other proteases, hopefully with the help of many of you in the audience. So we just recently, actually on Monday, I hit send on an NSERC application where we are applying this approach to all of the proteases in the coagulation system. And for NSERC purposes, we're doing this because we want to know more about these proteases and to maybe find evolutionary connections between these proteases based on substrate preference as opposed to based on gene sequence conservation. But really, we're hoping to build a new toolkit that will allow us to detect these proteases in complex biological mixtures like in blood or plasma. And some of you is around somewhere. Here's the plasminogen activation system. So we're doing this with UPA, TPA, and plasmid. And so hopefully we'll be able to build some tools that will be useful for looking at UPAR activity in non-cardiovascular disease settings. The other projects that we have going on with this sort of technology, I'll show a couple of slides about this, is thinking beyond just a deep characterization of a single protease. What about using this technology to profile all of the proteolytic activity in a biological sample in an unbiased way without any prior knowledge about what proteases may already be in that sample? And so as proof of principle for the application of this technology to detect protease activity in complex biological mixtures, we turn to the neutrophil. So some of you may know that neutrophils are the most abundant leukocyte in our blood, and they become activated in response to different agonists. And when they become activated, they release a host of serine proteases that play an important role in determining their response in our immune system, but they also release a number of matrix metalloproteases in which they play a role in tissue remodeling. And so we thought, well, would we be able to detect the individual signatures of each of these known proteases in a complex mixture like a neutrophil release agent? So we first started by doing a profiling of the two most abundant proteases in neutrophils, which is neutrophil elastase and cathepsin G. And now these proteases have been characterized by traditional phage display approaches in the past, and neutrophil elastase is known to prefer cleaving at valines and isoleucines, which aligns with the data that we see in our profiling data set. But of course, we're deriving these recognition sequences based on 6,500 cleaved peptides, as opposed to, maybe 2 or 3 dozen. And cathepsin G, on the other hand, is another serine protease that is a member of the chymotrypsin family and likes to cleave substrates after phenylalanine and tyrosine residues. So this told us that this profiling approach is widely applicable, can be useful in detecting the signatures of proteases outside of the coagulation system, and might be useful for this sort of broad utility that we envision for the future. So with that in hand, we then decided to take purified neutrophils. They're not really purified. In fact, we've got evidence to show that they're definitely not purified neutrophils on the next slide here. And what we did is we just activated them with PMA, which is a very strong antagonist of neutrophils, causes degranulation of their eosinophilic granules, releases all of the serine and metalloproteases into the buffer system. And when we applied our screening technique to neutrophils, we identified like 36,000 cleaved peptide sequences, which is an overwhelming amount of peptide sequences. In the presence of EDTA, which would chelate the active site zinc and inactivate all of the metalloproteases in the neutrophils, we didn't really see an appreciable decrease in any of the peptides that were detected. But when you used a broad spectrum inhibitor of serine proteases like AEBSF, we inhibited like 90% of the proteolytic activity in neutrophils. So this tells us that by far the most dominant proteolytic activity released from activated neutrophils comes from serine proteases, which is in line with what we know about the biology of neutrophils, thankfully. So then the next question is, so if we can use this technique in these broad, in these very diverse biological mixtures, what is the information that we can derive from those cleaved peptides? And so I've got a very talented undergraduate student, actually, who has been spearheading some of our deconvolution algorithm approaches to try and take those cleaved peptides and apply PCA and UMAP deconvoluting algorithms to try and break these cleaved peptides into categories based on amino acid property. And when we applied an early version of this to that 36,000 peptide sequences, we got about 8 or 9 clusters out of it. And many of those clusters aligned with substrate recognition motifs that are very reminiscent of what we expect for neutrophil elastase, cleaving at valine and isoleucine. Interestingly, we found serine protease activity that was not attributed to either chymotrypsin or elastase-like serine protease, but is actually more reminiscent to a trypsin-like serine protease, like those of the coagulation system. And what we think, these signals are coming from the fact that our neutrophil prep was just a sucrose gradient. So there's all sorts of other leukocytes that co-purified from that. And when we activated that cell mixture with PMA, we've also activated other cells to release their own proteases. So what I think this does is it shows that using technologies like this can be very useful in understanding in an unbiased way the way proteases are playing a role in these complex biological mixtures. And we're really excited about perhaps commercial applications of this, but also the general utility of this to explore protease activity in various fields. As many of you know, proteases play a critical role in cancer, bowel inflammatory diseases, certainly in cardiovascular disease, and also neurodegenerative disease. And so maybe using this technology to understand how dysregulated proteolytic activity can contribute to disease development, and maybe we could develop tools to diagnose or to detect these diseases early on in their progression. And so this is my call to anybody in the audience. We are seeking collaboration. So my lab approaches these questions from a protein biochemistry and a molecular biology perspective. We are not computational biologists, and so we are doing our best to come up with algorithms ourselves that will allow us to deconvolute these data sets. But this is certainly not our expertise, and we are looking for help. That being said, I hope that I've been able to convince you that being able to develop this technology might benefit your own field. For example, understanding how UPAR contributes to cancer metastasis. UPAR is the contributor to plasma activity in the coagulation system. And so maybe we can use these technologies to detect proteases in, CNS fluid, for example. And even if we can't precisely detect the protease that is responsible for the signatures, understanding how the signatures that we do detect change over time, or do these signatures, regardless of what protease they come from, differ between health and disease? That's an immediate diagnostic that we could develop to understand that disease. Okay, so that's sort of the first part of my talk, where I wanted to talk about protease biochemistry, and that's sort of, I would say, 25% of my lab is focused on developing that technology. The other topic that I wanted to discuss with you today, for those of you who think about genetics of complex or Mendelian traits, is this growing issue of how do we interpret genetic variants. So genome sequencing, exome sequencing, GWAS, these are great at statistically and unequivocally determining that this genetic variant is associated with disease, but there's very little information available as to whether or not that disease is causative or not, or is this sort of a bystander effect. And so this is an emerging issue. And so we're approaching questions about the interpretation of genetic variants with respect to von Willebrand factor. So VWF levels vary widely in the healthy population, and that's shown on the x-axis here. And individuals who have high circulating von Willebrand factor are at risk of developing thrombosis, and individuals who have low circulating von Willebrand factor levels are at risk of developing bleeding conditions. And patients with very, very low von Willebrand factor levels are actually diagnosed with this condition called von Willebrand disease, which is the most common inherited bleeding disorder in the population. Actually, estimates are at about 1% of the human population has von Willebrand disease. This is characterized by mucocutaneous bleeding, can cause post-surgical bleeding, dental bleeding, menorrhagia for women, and certainly postpartum bleeding is a huge concern for von Willebrand disease patients. And patients with von Willebrand disease are disproportionately are found with coding variants in the von Willebrand factor gene. But it's not known if those coding variants are causative for von Willebrand disease or if it could be caused by something else. And of course, this is something that many in the genetics community think about, that there is this emerging crisis of variants of uncertain significance, or VUSs. And the incidence of VUS has been steadily increasing in databases like ClinVar and ClinGen, and it leads clinicians wholly unequipped to manage patients who are identified as having a genetic variant in a gene that is known to be associated with perhaps illnesses. This is particularly the case in patients who are identified with a mutation in the BRCA1 gene for breast cancer, for example. But certainly that can be applied to other genes as well. And traditional tools simply do not scale. So, I'm a molecular biologist. I can make any mutation that you want, right? But it's like emptying a sinking ship with a teaspoon. You're never going to catch up, right? And so we need new tools to understand how genetic variation contributes to phenotypic changes in proteins. And this brings to this new technology that we've been applying in the lab recently called deep mutational scanning. So this is a technique where you build a library of every possible amino acid substitution at each position of a gene, and you push that library through some screening assay that will allow you to distinguish between variants of that gene that have an effect on its phenotype. And you can build these heat maps where you can identify mutations that are associated with gain of function activity or loss of function activity. And so many, many years ago, we applied this deep mutational scanning. Let me just go back. Outside of the human genetics component, from a protein biochemistry perspective, this is like alanine scanning mutagenesis on steroids. This is an enormous toolkit to understand how mutations can affect protein-protein interactions as well. And in fact, that is the way in which we approached our initial question using this technology. So we initially used deep mutational scanning in order to map the binding between ADAMTS13 and von Willebrand factor, knowing that ADAMTS13 requires these exocytes in order for substrate cleavage. We wanted to understand how is it that where are those binding sites and how important are they for mediating the cleavage of its substrate? So to do that, we turned back to our friend, the bacteriophage, and we used a fragment of von Willebrand factor that encodes this minimal substrate for ADAMTS13. It's a 73 amino acid peptide called VWF73. And we built a library actually just by mutagenic PCR. And in fact, there's more sophisticated ways of doing this now that I can talk about after the presentation if you're interested. And the idea is that when we expose this mutagenesis library to ADAMTS13, variants of VWF that harbor a mutation that impair cleavage by ADAMTS13 will remain uncleaved and associated with the bead. So what we did is we performed our screening assay at various reaction time points and pulled out the phage that remained uncleaved at each of those time points and looked for the gradual enrichment of mutations. And if those mutations were enriched with time, then we would know that that mutation is deleterious to its interaction with ADAMTS13. So we developed this heat map, which shows the impact of every amino acid substitution at each position of VWF73. And you can see that there's these very clearly demarcated zones where mutations have a severe impact on the ability of ADAMTS13 to cleave the substrate. This region here actually harbors the cleavage site. So ADAMTS13 cleaves between this tyrosine methionine cis-aryl bond here. And so it's not, perhaps not surprising that mutations around that cleavage site impaired the ability of ADAMTS13 to cleave its substrate. But we also found mutations in at least 3 other zones that seem to severely impact cleavage by ADAMTS13. And, we could spend, 20 minutes going through and looking at the amino acid property on this and the amino acid property here and make all of these inferences about how there's charge reflection and maybe cooperative binding and all these sorts of things. But I saved that for my graduate students who have to listen to me talk about this. But one thing that I will sort of bring your attention to, if we just look at the average impact of an amino acid substitution at a position of the substrate, we saw some very interesting patterns. So for example, the active site of ADAMTS13 is this really deep pocket, right? And you can see that consecutive residues around the cleavage site can be damaging to cleavage by ADAMTS13. Whereas these exosite binding regions around here, it looks like there's alternating sensitivity to mutations. And it turns out that these actually correspond to interesting structural features in the substrate. So for example, these exocytes map to disordered loops and beta sheets, which are known to have alternating 180-degree orientation of the amino acid with respect to its binding site. So what we think these alternating sensitivities mean is that there must be contacts between these particular amino acids and the protease, and we might be able to find corresponding pockets on the protease that are designed to accept these amino acids in particular. Whereas around the cleavage site, where it's this deep pocket, you could imagine amino acids in any orientation might be making important and productive contacts with the protease. And so we've been trying to work with structural biologists to take this information and do a simulated docking between this minimal substrate and the protease to finally get a structure of what that complex would look like. And we have not been successful yet. So if anybody wants to take on an interesting challenge, talk to me after the presentation. But, you know, outside of the sort of esoteric questions about protease biochemistry and protein-protein interactions, this is actually an important toolkit to the genetics community. And we are part of this growing group of enthusiastic scientists who are interested in applying these multiplexed assays of variant effect. So if this sort of research interests you, I would encourage you to go to the mavdb.org website. And there you can look at a list of nominated genes that people are either actively studying or nominating as high priority genes to be put through this pipeline. Or if, on the other hand, you would like to take on a project like this, you can register your interest in doing a project like that and seek collaborators in the community. And I can certainly put you in touch with people that are very, very good at doing this, and we're sort of plodding along in our own way. But we as an organization are making inroads to make sure that these data sets are clinically available to clinicians and can be used in the clinical interpretation of genetic variants that are constantly being uploaded to repositories like ClinGen and ClinVar. So that story was published many years ago, and so we've sort of been, as I said, plodding along in continuing to apply this deep mutational scanning technology to understand how genetic variation of von Willebrand factor can affect its role in the coagulation system. And we're focusing primarily on variants that may be associated with von Willebrand disease. So these are mutations in von Willebrand factor that lead to a severe loss in the circulating levels of this protein. And so we hypothesize that these mutations are preferentially impacting the biosynthesis and secretion of von Willebrand factor from the endothelial cell. And so we have an active program going on in the lab. We actually just got a great score on a grant on Monday to actually carry on this project, where we've developed this protein secretion assay to understand how genetic variants in von Willebrand factor affect its secretion from mammalian cells. So this has been the work of Taylor Sparring, who recently graduated with her PhD and is now working at AstraZeneca. And she validated a protein secretion assay, initially using GFP-labeled von Willebrand factor that's stably integrated into this engineered HEC293 cell line. And using variants of von Willebrand factor that are widely characterized to have impaired secretion, she initially showed that these variants lead to an intracellular retention of GFP signal and therefore von Willebrand factor. And so this was sort of the launching pad for us to do this unbiased mutagenesis approach, where we can take a mutagenesis library of von Willebrand factor and perform fluorescence-activated cell sorting and sort cells into bins based on the fluorescence intensity of those cells, hoping that mutations that are associated with a particular phenotype with respect to VWF secretion would be enriched in any one of these bins. So the way this works is Taylor spent 2 years building a mutagenesis library and then stably expressing them in this engineered landing pad, which was generously gifted to us by Doug Fowler and Kenny Matreyek at University of Washington in Seattle. And then you perform this functional assessment, and we're using FACT sorting, and then you just perform deep sequencing on each of the pools that you sort into and look for enrichment of mutations in these various bins. And so this is the initial heat map that she was able to generate, where she was able to show that, that every possible amino acid substitution at each position of this small region of von Willebrand factor can have varying impacts on VWF secretion from HEC293 cells. We identified about 140 possible mutations that lead to a severe retention of von Willebrand factor in these cells, and everything in between. The vast majority of variants, as we would expect actually from the heritability of complex traits like this, actually have no impact on VWF secretion from cells. And we even found some gain of function mutations in von Willebrand factor that can actually accelerate its movement through the secretory pathway of these mammalian cells. And so we have spent a couple of months now using these cell abundance scores and this deep mutational scanning data set to help us interpret variants that are identified in ClinVar and other like VWF-specific databases. And we have found that many of the variants that are anticipated to be deleterious or predicted to be deleterious do have high abundance scores, meaning that those mutations do cause an intracellular retention of von Willebrand factor. And then we've also been correlating those deep mutational scanning scores with clinical phenotypes of von Willebrand disease patients who have those mutations. And although the correlation for VWF antigen levels are quite poor, we did find that mutations that do not cause an accumulation of von Willebrand factor in the cell, those individuals have higher circulating VWF levels than individuals who have mutations that cause impaired secretion from cells. But what was very interesting was we saw a very strong and positive correlation between these abundance scores from the deep mutational scanning and the quantitative bleeding assessment score that patients with von Willebrand disease experience. So it does appear as though that there is some correlation between the ability of these mutations to cause an impairment in VWF secretion is associated with the severity of the bleeding phenotype that patients are experiencing who have these mutations. So, all right, I'm on time. So that sort of gives an overview of about 50% of what my lab works on. I spent a little bit of time talking about how we are using deep protease profiling to do this deep dive on individual proteases to sort of comprehensively define their individual substrate recognition patterns, but also thinking about using this in more complex and less well-defined biological environments. We've also got research in the lab that I just spoke about, looking at inherited traits and understanding how genetic variability in a protein can both inform some biochemistry about how that protein works, but also reinterpreting genetic variants that are identified from other omics techniques. And then about half of the lab works on ADAMTS13 as a potential therapeutic. So in my intro, I sort of talked about how VWF is this really sticky protein, seems to play a role in a lot of biological processes. And recombinant ADAMTS13 is like months away from being approved for the treatment of a very rare genetic thrombophilia. And we have been spending a lot of time in the lab actually engineering novel variants of ADAMTS13 to try and come up with generation 2 and generation 3 therapeutics, some of which we have patent protection for and we're interested in commercializing. So with that, I'll just stop and thank people who did the work. Taylor Sparring, who's now working with AstraZeneca, is responsible for the deep mutational scanning project. Rex Huang was a fourth-year undergraduate thesis student in the lab and did the neutrophil protease profiling project and is now back in the lab working on ADAMTS13 projects as a master's student. And, you know, every once in a while you get a gift from the heavens, and Enoch is that gift that we got. And, you know, we must have, I don't know what it was, but he called me as he was entering first year university and he wanted to have some research experience. And he's just been an absolute superstar doing all of the bioinformatics with the work that you've seen here. He's a third-year undergraduate student in the Bachelor's of Health Science program. So thank you very much for listening, and I'm happy to take questions.
Thank you, Colin. I mean, that was such a wonderful talk because I think it's actually really instructive to everyone about how to build a really impactful lab program, because you have really important biological questions that are centered in your own field of research interest. You built incredible technological platforms to interrogate those questions, but now you're recognizing those platforms actually open up more questions and more applications that can have much of a broader application. And that's what I find so exciting. So it's a really good time to come and talk to us about the intersection of some of the work you've done, but the platforms you've built that could be applied to our research questions too, right? So thank you. It was fantastic. I have a question about protease binding partner fidelity. And I'm wondering, because I'm not a protease biologist by any means, but I'm wondering about context specificity in terms of some of your platforms. You mentioned the cell systems that you've chosen to interrogate, like neutrophils or so on. Would you expect that you might find different binding partners depending on tissue specificity or the context of different cell types? And if proteases are promiscuous, then what we find in studying cancers is a lot of times something has one binding partner in a normal tissue context, and in the neoplastic context, it has yet again different binding partners. So, have you started to explore some of that context specificity?
Yeah, so we actually think about this quite a bit. I think this is where having fundamental training in biochemistry can be really helpful. Understanding what a KD value is, right? Understanding that the strength of a binding interaction is not just an inherent property of the protein, but is dependent on the relative concentrations of the two constituents. So pay attention in your undergraduate biochemistry projects, because these are the sorts of things that make all the difference in biological studying. So for example, the matrix metalloproteases, which are really important in tumor metastasis, they have incredibly dense substrate availability when they're combing through the extracellular environment. And so their relative catalytic efficiencies can be quite low compared to other proteases, like in the coagulation system, where it's a little bit more diffuse. And so building tools to interrogate the specificity of metalloproteases is traditionally quite challenging because of that. And this is where we think maybe a platform like this can be useful, because we could look at the entire substrate space that could be available to that protease and make tailor-made substrates that can work in an environment that you are capable of pulling that protease out of.
Because people have only found the traditionally the strongest binding partner
That's right.
and the most frequent binding partner, but
That's right.
maybe ignoring other substrates.
Yep.
I'd like to open to questions. Who has some questions for Dr. Kretz? Everyone's deeply thoughtful right now. And by the way, you're speaking to most of the students here are enrolled in biochemistry. So I think they're all smiling and pretty happy about that. So
Chair of biochemistry.
yes, yes. Daniel.
No. Is the short answer. I dream about a lot of things, but I haven't dreamt about that yet. Doesn't mean it's not possible. In many ways, that's like an engineering challenge, and nobody would have thought that spatial transcriptomics would ever be a thing, but the engineers figured out a way to develop these chips where spatial transcriptomics is now definitely feasible and starting to be widely used. So I imagine that we could envision a scenario where we could apply this eventually. But I think we have enough challenges on our plate right now using this in a simple platform that once we get that sorted out, though, that's a great idea.
Not to mention that you can tell that Daniel comes from a solid tumor background, but Dr. Kretz's tissues are all liquid, right? So I don't think that cytoarchitecture is quite as important for some of these questions as it is for us.
Sure.
But I think Daniel, who studies cancer metabolism, is thinking about the fact that I just came back all stoked from a conference, because now they're actually doing spatial metabolomics.
Oh my God.
So like it's just gonna be there's always these next frontiers, right? So I think it's a great idea as well in terms of thinking of a spatial application for this platform. Yeah. Please.
Yeah, I think there's only so many questions we can answer at a time. But that's not to say that it's not possible. So people have been using deep mutational scanning, for example, on yeast cells, and people are even doing mammalian cell for deep mutational scanning. It's not hard to imagine a way to adapt our protease technology for that as well. But this is certainly something that as a platform is starting to gain traction in a lot of different arenas. I don't know if it's quite that I've seen yet made it to the glycomics world, but certainly, you know, you could imagine doing a deep mutational scan for lectin binding on a display protein. Like these are certainly things that are in the realm of possibility. Yeah, it's a great idea.
This concept where you find that hydrophobic pocket.
Yeah.
That opens the protease.
Yeah.
This is pretty rare. That's a powerful thing to explore.
Yeah. So, you know, I didn't talk about, and I probably should have talked about this to a cancer group, but, you know, ADAMTS13 as a metalloprotease is the only metalloprotease I've ever encountered that is not inhibited by those small molecule MMP inhibitors, metimistat, betimistat, ilemistat. All those drugs that were pulled out of clinical trials because of toxicity issues don't touch ADAMTS13. In fact, that was part of our JTH papers. We made mutations in ADAMTS13 to try and sensitize it to these things. And we had to jack that protein up in order to get it inhibited by these broad spectrum inhibitors of metalloproteases. And we do think that it's due to that hydrophobic pocket where that pocket is collapsed. And the substrate, I didn't talk about it, but the substrate is a very, very greasy substrate. The cleavage site's buried, actually put a slide for it. Might have got a question. But the cleavage site's buried right in the hydrophobic core of this domain, is not normally accessible to the protease. And you actually have to pull on that domain and unfold it by force. It takes 10 piconewtons of force to unfold that domain and sensitize it to cleavage. And so because of where it's located, it's a very greasy substrate. And so the protease itself would have had to evolve and adapt to that for its substrate. And that, of course, would not be stable to an aqueous environment. So what we think is happening is that you've got this greasy hydrophobic pocket that's protecting itself from the aqueous environment. And when the substrate becomes rapidly shear activated, it sort of slides in and conformationally activates the protease. So that's the mechanism that we're sort of trying to explore right now in the lab biochemically, and try and prove that without a shadow of a doubt. But you're right, I can't think of any other protease that, you know, there's these rhomboid proteases that cleave substrates in the fatty pocket of a phosphatidylserine membrane. That's where they cleave their substrates. So maybe some of those would also have this kind of function, but nothing certainly in circulation. Which
that targets that.
Yeah. That's right. Well, and we've shown that once you've activated the protease, it is promiscuous. So that could be an interesting biotechnology application. Yeah. Yeah. Great idea. We should talk.
Any other questions for Dr. Kretz? Okay, so otherwise help me please thank him for a fantastic talk.